In our previous lecture, we discussed about interphase, which is actually a preparation stage for the mitosis. Now we are moving towards the next phase of the mitosis, which is termed as prophase. Okay, we are going to discuss the key changes which are taking place during the prophase stage of the mitosis. Always remember, in the interphase, the DNA was loose and spread out in the nucleus. You can see here. Okay, here all the DNA was in the form of loose thread-like structure, which is termed as chromatin. Okay, but as we move towards the prophase stage, let's zoom into prophase stage. You can see now the chromatin is in the form of highly organized structure, which is termed as chromosome. How chromosomes basically look like: each chromosome consists of two sister chromatids. You can see here, these are the two sister chromatids. Okay, and these two sister chromatids are connected at a center point, which is termed as centromere. This is also an attachment point for the spindle fibers. So the first key change that occurs, that the chromatin or the DNA, which was previously in the loose thread-like structure and was dispersed inside the nucleus, is now becoming highly organized, is converting into the highly organized structures which are called as chromosomes. The second important thing or change that happens during the prophase is that now the chromosome starts becoming more shorter and thicker as prophase progresses. Okay. The third important change that is going to take place here is your nucleolus is going to disappear. As you can see in the interphase, this nucleolus was present, okay, but as the prophase starts, the first thing that disappears is the nucleolus. The fourth important change that is going to take place is the disintegration of this nuclear membrane. Okay, you can see now the nuclear membrane is breaking down into different pieces. Okay, so you can say that nuclear membrane starts breaking during the prophase of the mitosis. Now the fifth important change that is going to take place is that the position of these centrioles. Okay, previously in the interphase, these centrioles were lined up very close to each other, but now you can say that these centrioles are moving towards the opposite poles of the cell. Okay, once they reach at this position, okay, they are going to generate specialized structures which are termed as spindle fibers. So not only centrioles are rearranging themselves and moving towards the opposite poles of the cells, but also they are going to generate the spindle fibers. So spindle fibers begin to form at the end of the prophase. So you can say that prophase is all about that the cell is preparing the chromosomes for the proper movement during the further stages of the mitosis. So you can always remember prophase from P as packing phase, because everything is packing here and preparing itself for the next division, which is the metaphase. You can remember it as PMAT, that is prepare, please make a table. What does that mean? When you say please make a table, it means that please arrange everything before we can eat. Okay, so here the cell is actually arranging and packing everything before actual divisional process starts. Now let's move towards the third phase of the mitosis, which is termed as pre-metaphase. Actually, the pre-metaphase and metaphase, they are the two key substages of the same stage, that is the metaphase. Okay, so first a short stage exists which is termed as pre-metastage. Let's discuss this stage. So what happens in the pre-metaphase stage? You can see that here the nuclear envelope or nuclear membrane, which was previously present in the center of the cell, has now become completely disappeared. So you can say that nuclear membrane become disappeared in the pre-metaphase stage. Okay, so now you can see that all these chromosomes are free to move inside the cytoplasm of the cell, and they are now actually become quite scattered in the cytoplasm of the cell. Okay, Also, you can say that the centrioles, which were moving towards the opposite poles in the prephase, has now reached the opposite poles. Okay, so now centrioles have reached the opposite poles, and now they have, sorry, now they are preparing properly the spindle fibers. Okay, so spindle fibers, you can see these are the long thread-like structures in the white color, which have been originating from these centrioles. Okay, so here ends the pre-metaphase stage. So let's move towards the metaphase stage of the mitosis. Okay, so as previously, you can see that the chromosomes which were previously scattered inside the cytoplasm of the cell, now in the metaphase all the chromosomes line up exactly in the center of the cell, and this center line, which has been prepared by these chromosomes, this line is termed as metaphase plate. So you can say that in the metaphase, the chromosomes line up in the center of the cell, making a plate which is termed as metaphase plate. Okay, now the spindle fibers which were scattered in the pre-metaphase stage, now they become more aligned and start moving towards these chromosomes. Okay, because there is a protein which is present here in the center region of the chromosome, which is called as centromere, and these spindle fibers are going to attach to that part of the chromosome which is termed as centromere. Okay, so you can see that, you can say that the spindle fibers starts attaching towards the centromere of the chromosomes, and this is very important checkpoint stage of the mitosis, because every chromosome should be properly aligned at the center of the cell. Okay, and everything should be in proper balance so that the spindle fibers can properly attach at the centromere of the chromosome. If for any reason the alignment is wrong, the division will not proceed. Okay, so for example, the chromosomes have aligned themselves in such a position that they are not lined up to make the metaphase plate. Okay, in that condition there is no separation of these chromosomes, because there is no proper metaphase plate formation, and spindle fibers will not be able to attach to the centromere part of the chromosomes. Okay, so this is actually a decisive point that whether the mitosis is going to take place or not. Okay, and for the mitosis to take place, the chromosomes should be properly aligned at the center of the cell, thus making a metaphase plate. So metaphase, you can say, is like the students which are standing in the straight line before any particular race starts. Okay, so you can remember metaphase as M equals to middle, because the chromosomes are going to attach in the middle of the cell, are going to arrange in the middle of the cell during the metaphase. So what is prophase? Prophase is actually the packing stage of the cell. Okay, and what is the metaphase? Metaphase is actually the meeting in the middle stage of the cell, where the cells chromosomes arrange themselves in the middle of the cell. Okay, so let's quickly review prophase. So in the prophase, the chromosomes become more and more visible, they become short and thick, nucleolus disappear, nuclear membrane starts breaking. Okay, and the spindle fiber starts originating, not properly forming. Okay, just starts originating. Then there comes a pre-metaphase stage, where the nuclear membrane completely disappeared and the chromosome started scattered in the cytoplasm. Also, centrioles reach at the opposite poles of the cell. What happens in the metaphase stage? Chromosomes get themselves properly arranged in the middle of the cell, thus making a plate which is termed as metaphase plate. Also, the centriole starts making spindle fibers which are not going to attach to the centromere of the chromosomes. Okay, so in our next lecture, we're going to discuss about anaphase and telophase. I hope you have understood these four key stages of the mitosis, that is the interphase, prophase, pre-metaphase, and metaphase. Thank you.